Hi, everyone. Happy Friday to you. I hope your second week of classes has been going smoother than the first. At least for me, it has. So I'm kind of happy about that. But I'm still very glad that the weekend is here. All right, let's go ahead and uh, continue talking about our innate immune system. Last couple lectures, we were talking about how our innate immune system, the part of our immune system that we depend upon like 99% of the time on a daily basis to deal with common everyday pathogens and threats, how the innate system itself kind of has two parts to it. There's the cells and proteins that are in charge of immediately dealing with things. The moment you get a cut or you're poked by something or there's a breach in your skin or the lining of your GI tract and microbes come in, that's the moment that you're experiencing infection. You have your resident cells, meaning the things that are already there, like macrophages, that can go to work, right? And you have proteins like the complement proteins that are already flowing through your blood and seeping into your tissues that can also go to work right away. And so that's our immediate response. And most of the time, that's good enough, right? I mean, you get paper cut or a thorn or something like that. A few hours later, maybe you don't even notice anymore, right? Because the immediate part of your immune system took care of things really well. However, if in reality the invasion by the microbes is a bit greater, like there's more bacteria or virus, or those pathogens are a bit sneakier, they're having a harder, your immune system is having a harder time doing things like complement proteins trying to coat them and macrophages trying to eat them, and that's not working out so great. Your macrophages will need to call for backup. And that backup is what we would call our induced response, right? It happens after a little bit of delay, but it's still part of our innate immune system. So that's what we're going to start talking about today and finish talking about it on Monday. Okay. So let's kind of think about what's going on. The immediate part of our immune system means within the first few hours, right, we have pathogens that invade the tissues and they begin to proliferate, and they begin to multiply. Fortunately, the pathogen is recognized by preformed, ready to go, soluble molecules like our complement proteins and also our resident effector cells like our macrophages that are already there patrolling around. The pathogen is eliminated, the infection ends, and that's the end of the story, right? You get a paper cut. Most of us, when we get a paper cut, by the end of the day, we're not even bothered by it anymore, unless it's in a particularly annoying place, like on your knuckle of your finger or something like that. So there's very minor tissue damage, and that's just repaired, and then that's it, right? You don't even necessarily have any leftover evidence that you had a little paper cut. However, so let's look at this. If you have a pinprick, right? So this would be an example of a very sort of minor wound. This is an electron micrograph really zoomed in at the head of a pin. It's so zoomed in that you can actually see, right? The head of the pin isn't really sharp at all. Well, I mean, it is sharp in the sense that even though it looks rounded in this, the point is that this zoomed in little point is actually still way smaller than 2 cells in your body. So this is still small enough to pry its way through 2 cells of your body so it can actually poke its way into your skin, right? But look at these little yellow things on it. What are those, right? Those are bacteria, right? So a typical head of a pin has thousands of bacteria on them. So that means the moment this pin goes into your finger, like the picture here, of course, these thousands of bacteria, right? They're like, yay, right? They're in your tissue. They come off the pin and they start doing their little thing to make themselves at home, right? But fortunately, we have our effector cells, like our macrophages, that are patrolling around in there. So as these bacteria are coming into your tissue, your macrophages start picking them up, right? And you have complement proteins helping out, right? So the complement proteins are helping out by opsonizing the bacteria, like in this cartoon here. And remember, this is not drawn to scale, right? It's not like you only have 8 lollipop-like complement proteins stuck on a green pill-like bacteria, right? In reality, you got millions of complement C3 coating the surface of this, opsonizing it to make it easier for the bacteria, for the macrophage to grab onto. And as we learned last time, these complement proteins can also initiate the C5 membrane attack complex pathway. And these two things are happening at the same time. Right? Because if you have millions of these C3b molecules on there, some of them are continuing just to spread and put more C3b on. Some of them are doing the membrane attack complex. So that's all fine and good. And that's what happens so that you have this pinprick, and by the end of the day, you're done, right? I mean, your immune response is done. It's succeeded and you're fine. However, pathogens might be able to outrun this immediate response. For example, if there's too many microbes that came in, or the microbes are poorly recognized by your macrophages, by your complement proteins, they're having trouble doing things like binding to it. And also some microbes have evolved to fight back and resist things like the complement proteins, and resist things like cells coming to attack them. So in this case, the bottom line is the immediate innate response can't keep up and the pathogen is not eliminated. And so time is going by now. So for example, this is if you have a large wound, so a lot of bacteria can get in, or if you're talking about a wound, something like the lining of your intestines, where suddenly you have a whole flood of bacteria coming in. Remember, the commensal good bacteria that's in the lumen of our gut, helping us with digestion and things like that. The moment they enter your tissues through a breach in the cell wall, they become pathogenic, right? Because they are now perfectly happy trying to set up home in your body's tissues where they don't belong. So now the induced response has to come. This is the backup. So now we are talking about maybe from 4 hours to 4 days, right? So again, the pathogen's invading the tissues, but the activation of the resident cells and the complement proteins, maybe it's not enough. So you begin to get these telltale signs of the induced response, including inflammation, things like fever, and a process that we call the acute phase response involving the liver, which we will look at today. So the next step then is these will result in more molecules, effector molecules, and more effector cells, not necessarily just macrophages, but things like neutrophils become mobilized, and they start helping out at the site of the infection. So after maybe 3 or 4 days, then your innate immune system succeeds, right? And the pathogen is eliminated and the infection ends. But now, as a consequence of the delay and the fact that the pathogen was allowed this extra time to cause damage, you're going to have greater damage, right? So that's why you have maybe something like a scar or some other type of tissue damage. But, that's part of your tissue repairing itself. Let's look at some of these symptoms of the induced response. Inflammation. Right? If I say, what's inflammation? I think all of you would kind of come up with some maybe general terms for inflammation. That's like swelling, tenderness, redness, that feeling of the tissue being particularly warm. If we're talking about a cut in your skin, right? That's inflammation. So why is inflammation important? It's important because the inflammation that you experience, right, like the swelling, the firmness, the sensitivity to the touch, the warmth, right? These are actually things that your immune system is doing. It's not like the bacteria itself wants to cause inflammation, because why would the bacteria care if your skin gets warmer or the blood flow is being altered and things like that, right? These are all responses, physiological changes in your body's tissues meant to help fight against the bacteria. This can also include things like pain, right? The sensitivity or maybe overall sensitivity, like you suddenly get some aches and things like that. Fatigue. Fatigue is an interesting one. Our immune responses, when they are mobilized, absolutely are drawing upon different resources, different energy from our body. And that comes as a cost, right? Because if you're trying to still function normally, and your body's trying to battle these these microbes, right, the most important thing is you defeat the microbes, right? So evolutionarily, oftentimes we get tired as part of the induced response, because if we just lay down, rest, and maybe sleep, that allows our body's resources to be diverted towards fighting the microbes, right? And fever. Fever is a part of this because the raising of the tissue temperatures, like our body's temperature above the normal 98.6 Fahrenheit or 37 degrees Celsius, is a way to actually change our tissues to be uncomfortable for the microbes. The microbes have evolved to expect 98.6 Fahrenheit, and so they might thrive best at normal body temperatures. So if your body raises its temperatures by a couple degrees, 99 or 100-degree fever, right, that might slow down the bacteria from multiplying, right? So fever, again, we all know what fever is. We've experienced lots of times when we've had fever, but a lot of times you don't think about it. You blame it on, oh, it's because I have an infection, so the pathogen is giving me a fever, when in reality, it's your body is giving yourself a fever in order to fight back against the pathogen. So when you hear these descriptions, you realize, yeah, already we're saying that this induced response that involves these things is now more expensive than just the immediate response, right, of just having the macrophages and complement proteins take care of it. It's not nearly as expensive as the full-blown adaptive response, which we will learn about in a couple lectures and spend most of the semester talking about. That's the SWAT team, right? But still, this is getting more expensive. That's why it's sort of a delayed response of your body. In order for macrophages to be involved in this, because after all, it's the macrophages and their activity that's going to lead to whether the induced response starts happening. The macrophages are busy eating things like the microbes, and as we began talking about, these macrophages are releasing small proteins that are meant to be messages for other immune cells to start mobilizing. Right? These are the small cytokine messages. I think on the cartoons, like in the previous one, they're often represented as little colorful triangles just to show that they're simple. Right? So how do the macrophages even really detect that there are pathogens present, right? Either directly when they're contacting a bacteria, or how do they detect that there's the presence of bacteria, like different types of things that are being released by the bacteria? They do this through a series of different receptor proteins on their surface. So the surface of a macrophage, the cell membrane, just like every other cell in our body, right? It's a lipid bilayer, phospholipid bilayer, but there's all kinds of goodies kind of embedded in that layer, including a whole bunch of different proteins. And these proteins are different shapes so that the shapes can help them interact with different things that are coming from the outside and arriving at the surface of the macrophage. So there are over 100 different receptor types of proteins that are involved in immune cells. And each type of immune cell that we will talk about over the semester expresses a subset of these different proteins. Remember, the genetic information, the genes that code for these different proteins are all there in your genome already, right? In fact, not only every immune cell, but every cell in your body that has a copy of your genome in it has these genes in it that code for these particular immune receptor type proteins. It's just a matter of whether that particular cell type bothers to express those genes to make these proteins as part of the normal cell's sort of purpose or way of functioning. Right? So different immune cells will express different subsets of these immune receptors, and that's all part of what they're supposed to do in order to help them do their job. This variability allows for many different types of populations of immune cells to each be kind of more specific in detecting certain types of pathogenic evidence or features. And therefore, overall, your immune system, right, has a stronger ability to detect a diverse range of different things being presented by the pathogens. Oops, sorry. Before we move on, so here are just some examples of main sort of classes of macrophage receptors that are in charge of recognizing components that are coming from the surfaces of microbes. We already looked at complement receptors, like this little blue one in the middle. This is like CR1, for example, right? Complement receptor, it's designed to be able to detect complement proteins like C3b that are opsonized or fixed on the surface of a bacteria, right? But in addition, here are some other examples. We have mannose receptors, right? Both of these, this big tall mannose receptor and this short dectin, right? These are receptors that are the right shape to detect certain sugar structures on the surfaces of microbes. Right? Then we have other ones like this Marco SRB, right? These can detect other types of molecules that are released by the microbes. So let's take a closer look at these just few examples so you have a sense of how they work. Receptors like the mannose receptor, like the dectin-1, fall into a big family of receptors that are known as lectins. So lectins are proteins that can bind to sugar groups, right? So they don't have to be proteins that are a receptor that are stuck on the surface of a cell. It could be a free-floating protein that binds up sugars. They're all called lectins because they bind sugars, right? So they are receptors for carbohydrates. So macrophages use these lectins to bind certain types of sugars that are on the surface of bacteria that show up a lot more on the surfaces of bacteria than the normal healthy cells of our body. Okay. Mannose is a good example. This is the mannose sugar right here. It's very similar to glucose, right? But it is chemically different. Mannose is very abundant on the surfaces of many pathogens, like on the surfaces of the cell walls of certain bacteria. So it would make sense then, if this is kind of like a generic feature of many bacteria, this would be like the rainbow wig on the normal everyday clown, right? The generic clown. So evolutionarily, it makes a lot of sense that our immune systems, as part of its innate response, comes pre-equipped with these little tools to detect the presence of mannose, right? Mannose is far less common on the surfaces of our normal body cells. So again, that makes sense, right? So if a macrophage detects another cell that has a lot of mannose on it, right, evolutionarily, it's a safe bet that this is something that doesn't belong in our body. So dectin-1 is probably the most primitive type of lectin receptor. This is just a cartoon that shows the cross-section of the cell membrane. So this would be the membrane of the macrophage. This protein, this dectin-1, again, right, very oversimplified because it's just showing you functionally. There's a part of it that sticks through the membrane and keeps it anchored there. There's a part above it that acts as the part that actually is the right shape to fit something like mannose sugars. And there's a part of it that sticks into the cell, the cytoplasmic domain, that actually transmits the signal that this receptor has bound to mannose, right? So if something binds here like mannose, it causes this protein to change its shape in order to get more comfortable, right? The polypeptide chain of the protein has to shift around a little bit because now you have this sugar stuck on it, and you get a ripple effect, right? As that part of the protein has to change, there's a ripple effect. The other parts of the protein have to shift around as well, and that will change the shape of the cytoplasmic domain so that other proteins inside of the cell that might ordinarily just float by because it's the wrong shape for them to act with, suddenly the dectin cytoplasmic domain is now the right shape, and those proteins that are floating by suddenly interact, and it can actually coordinate different proteins now fitting together, activating those proteins, and those proteins then talk to other proteins in a big chain reaction that eventually lead to the signal getting into the nucleus of the cell and maybe triggering the cell to start expressing certain genes like cytokines that up until now, those genes were turned off, right? So dectin is a very simple one. Mannose receptor is a bit more complicated. You can see the mannose receptor actually has many of these mannose binding domains, each one of these green Pac-Men, right? They can bind a mannose. And that means it's more sensitive to binding multiple mannose sugars on the surface of a bacteria that might be pressing up against this. Keep in mind, of course, that this is just one protein. So compared to this one protein sticking out from the surface of the macrophage, a bacteria that's coming by, it would be like a humongous blimp, right, in comparison. So this would just be pressing up against the surface of the bacteria, and these mannose binding domains have a chance to detect the mannose. This on the end is what we call the ricin type binding domain. It makes the whole thing look like it's like a cartoon flower, like a rose with petals, right? What is this ricin type binding domain? The shape of this region of the protein bears a striking resemblance to the protein ricin, right? That's why it's ricin type. What is ricin? Some of you may have heard of ricin before. Ricin is a highly toxic protein. In fact, it's one of the most toxic proteins that we know of. The reason it's toxic is because it's made of two parts. So this ricin protein is two peptide chains represented. One is brown here, one's purple, right? And when they fold up together, they form this one ricin molecule. One part of this, either the blue or the purple, either the purple or the brown, I don't remember now which one it is actually the right shape to bind to the surface of your body's cells. Okay, and so it binds to these galactose amines that are found on the surface of your cells. So your cells have galactose sugars on them. That's what helps get this ricin protein into your cells. And inside of your cell, the ricin protein falls apart. And the other part is the dangerous part, because the other part is the right shape to bind to certain features of your ribosomes. Remember, the ribosomes are the factories inside of your cytoplasm that are busy doing what? Busy translating messenger RNA into finished protein products that your cell depends upon. Right? So there isn't a single moment of any single day of your life when your cells are not making proteins, right? And it's the ribosomes that are doing it inside the cells. This ricin molecule comes and it gums up the ribosome. It prevents the ribosome from continuing to work on mRNA, right? And as a result, your cell can't make any proteins from that ribosome. Right? That's bad news. They've actually done studies that showed that one of these toxic ricin molecules, it'll mess up a ribosome, stop that ribosome from working, it'll fall off and can go to another ribosome and stop that one, right? They've actually done studies that showed that one ricin molecule entering one cell can disable 1,500 ribosomes per minute inside that cell. There's no way the cell can survive that, right? So that's why ricin is so toxic. You may have heard of ricin because, unfortunately, aerosolized ricin has been used in the past as terrorist attacks, right, in the gases and things, and it's highly toxic. So why would we have something that looks like ricin on the surface of the receptor? Because this part of the molecule here is the shape of the ricin that's only involved in binding to the galactose sugars, right? The other part, the dangerous part of ricin, is not here, all right? So what that means is that as your macrophage is pressing up against the bacteria, you can imagine then that this receptor would kind of bend over, right, as it's getting squashed, and these green Pac-Men then have a better chance then of binding to the mannose sugars on the surface of the bacteria. Eventually, this thing bends over, and the ricin part will actually attach to the surface of the macrophage, again, forming like this little upside down U, like a little bridge. Why is that important? Because that helps secure the receptor, anchors it even tighter as this big blimp of a bacteria is pressing down on it, right? So it's really cool to think that this protein has evolved to have that ability, on the one hand, to start off kind of flapping in the wind, because that's how you increase the ability for it to detect anything. But after it's been kind of pressed down, all the little mannose binding domains have had a chance to latch onto something, then you really anchor the receptor so that now the macrophage has a good chance of holding onto the bacteria tighter, okay? What about these other receptors? Another very important immune receptor that comes up time and time again in discussions of how our body responds to things like bacteria are these toll-like receptors, T-O-L-L-like receptors. These receptors are in charge of picking up certain lipopolysaccharide molecules, or LPSs, that are coming from the bacteria. What's an LPS? So many bacteria have thick lipopolysaccharide coats on them. I already showed you examples of this early on. This is like the shag carpet, right? The shag carpet that's on the outside of the bacteria that helps hide the presence of other protein features on the bacteria, making it less of a target for things like macrophages to normally be able to grab onto. Right? These lipopolysaccharides, they are there's a little fat molecule with a long string of carbohydrate. These oftentimes fall off of the bacteria. It's just like a shedding dog, right? Like you go over to your friend's house and she has like this big shaggy dog and her whole like sofa is covered in dog hair, right? So bacteria are like that too. They're like shaggy dogs that are shedding LPS. Since LPS is a very common generic feature of many bacteria through eons that they've evolved with us, again, this is a good signal that our immune system is built in to recognize. So immune cells have these toll-like receptors. Here's the bacteria, here's the LPS molecule that's shed. So you have proteins called LPS binding proteins that are floating around. They're the right shape to bind to an LPS molecule, and they are also the right shape to attach to this particular receptor on the surface of the macrophage known as CD14. And so when they attach, they pass the LPS molecule onto the CD14, and that CD14 in turn associates with another molecule called MD2 here. That's part of this big receptor structure. So this yellow part of the structure is the actual toll-like receptor, right? And once the MD2 is activated, because this lipopolysaccharide has been given to it and it changes its shape, right? That causes the toll-like receptor to change its shape. And just like I said before, with other receptors, the cytoplasmic part of these proteins then change their shape, and suddenly proteins that are inside the cell pay attention, can interact, and you kick off a chain of proteins talking to each other that result in the macrophage turning on certain genes. So here's the actual structure of the toll-like receptor. And so you can see the gray part here is the toll-like receptor. In this particular case, toll-like receptor 4, or TLR4. The blue is the MD2 that received the lipopolysaccharide, and then the red is the lipopolysaccharide itself. Right. So LPS binding to the toll-like receptor activates the cytoplasmic ends of the TLR4 to cause signals then to go into the cell and change the cell and activate it. Right. So this name toll-like receptor, where does that come from? Right. A lot of proteins in our bodies were first identified in simpler organisms, such as Drosophila, fruit flies. Right. I think all of you know fruit flies are a very popular animal model or insect model for studying things like genetics, because you can cultivate them, and if you make mutations in them, you can study when they breed new flies. Generations happen very rapidly, so you can actually see the effects of mutations and things like that. So the original toll receptor is the one that was identified in Drosophila, in the flies. And as the story goes, it was first discovered by a German bioresearcher, and her interest was in understanding how different proteins in the Drosophila, right, on the surfaces of the cells, were related to changes in cell function. And so she discovered that this particular protein receptor on the surface of the fly immune cells, when it was stimulated, caused a dramatic change in the immune response, to the point where she exclaimed in German, "Das ist der Toll," right? I know my German's horrible. Any of you who really speak German, you're probably like, what? But she said, that's amazing, right? And so Christian Woldhart, the researcher who discovered it, named it the toll receptor in flies. So we have a similar receptor because evolutionarily it's conserved, right? What worked in insects, many of these common types of proteins are carried on through evolution, and we as mammals have it. Our version is slightly different. That's why it's toll-like, right? Toll-like receptor. All right, so how does TLL4 mediate the expression of cytokines? So here we have the first panel. Here is just like the previous slide. The LPS that's being shed by the bacteria is picked up by proteins that help get it to the TLR4 receptor, right? Here that changes the shape of the domain that's in the cytoplasm of the cell. So proteins that were floating by before and not paying attention because it's the wrong shape, suddenly the TLL4's cytoplasmic domain is the right shape for proteins such as MyD88 to come along and attach. Right? When MyD88 attaches, right, you start recruiting other proteins, like, for example, this IRAC4, TRAF6. It kicks off a whole bunch of proteins talking to each other, which we call a kinase cascade. One protein passing on phosphate groups to other proteins, changing their shape and activating them. And eventually through this chain of proteins interacting with other proteins, it activates this very important transcription factor protein, NF-kappa B. Right? NF-kappa B is a very primitive protein. We see it very early on in the evolution of complex living organisms on the planet, because NF-kappa B then, when it's activated, goes into the nucleus and it attaches to the regulatory sites of DNA surrounding genes. So things like enhancers or promoters of those genes. And therefore, a gene like represented here that was maybe silent before then gets activated. NF-kappa B turns on the transcription of this gene by recruiting other transcription factors that then result in RNA polymerase, RNA pol2, transcribing this. And so then you have these messages, right? These are now RNA messages that then get processed right through RNA editing. And they are then translated by the ribosomes, right? These green things into the finished cytokine proteins, right? And these cytokine proteins, they go through the endoplasmic reticulum and the Golgi complex, they get packaged, and then they are released out of the macrophage. And these little cytokine, these little triangles then are the call for backup, right? So other immune cells that have the right kind of receptors on their surface, cytokine receptors, can pick up on this message and start mobilizing. So how much of this do I want you to know? I don't require you to memorize all of these intermediate proteins, right? Because at the level that we are discussing this, it would just be you memorizing a list of names. And to me, that's not that important. What I do want you to understand, though, is what's going on in the first panel here and how that leads in the second panel to this important protein, MyD88, suddenly being involved now. And you can skip ahead. I want you to appreciate that this chain of events here leads to the activation of NF-kappa B. And that's why NF-kappa B, we'll see NF-kappa B again, is a very important transcription factor in immune responses, right? Because it helps turn on things like cytokine gene expression. Okay. So now we are getting into this induced response then. These cytokines, the little triangle here that's being released by the macrophage as a result of it detecting the presence of bacteria, right? More and more of these little cytokines are being released, okay? After maybe a few hours, right, the level of cytokines are building up in your blood and it's being received by other cells. But if your immediate response, the macrophages, the complement proteins, succeed in eliminating the bacteria after just like 4 hours or so, then you would imagine the levels of cytokines being released into your blood drop, right? So you never really get the induced response, the backup response, right? However, if the macrophages after a few hours keep eating bacteria, right, more and more cytokines are being released, the cytokine levels begin to build up to a point where now really you start seeing a lot of cells responding and coming to help out. And that's your induced response, right? That includes things like recruiting more monocytes that are floating through the blood to stop and leave the blood vessel to become more macrophages, and the recruitment of neutrophils. Okay. Neutrophils, let's talk about these. So neutrophils are still part of our innate immune system, but they're very much part of our induced response That's brought on by macrophages calling for help. We have tons and tons of neutrophils in our body. Remember, when I said the 5 types of white blood cells that are floating around in your blood, and you were to say use the never let monkeys eat bananas, neutrophils is at the top. 50 to 70% of the white blood cells in your blood are neutrophils, right? Amazingly, that's even a small part of the sheer number of neutrophils that are hanging out in your bone marrow, waiting to be released as part of this induced response. Neutrophils are very powerful phagocytic cells, right? They are much more efficient at eating things like bacteria compared to something like a cumbersome macrophage doing it. So you can see here, as part of the induced response, cytokine levels are building up in your blood that triggers your bone marrow to release tons and tons of neutrophils. The neutrophils start flowing through your blood, all through your circulation. They get to the site of infection, which is shown here, right? And then they make their way out of the blood vessel. So this process of immune cells squeezing their way out of the blood and into the surrounding tissue is a process that we call diapedesis. It's a weird word, okay, but we will look at that in greater detail. And the neutrophils then join the fight. Okay, so now you have the macrophages that are already there and have been there all along trying to eat things, right? But now all of a sudden you have the backup. The neutrophils come in, and you can see the neutrophils are very effective now at eating up far more of the bacteria, right? The problem is the neutrophils, right? They're so powerful as a phagocytic cell. Part of that is because when they eat something like bacteria, right, the destructive power of the neutrophil on that bacteria is so great that the neutrophil itself can only last for a few times of eating bacteria. A typical neutrophil might eat just maybe a handful of bacteria, maybe 3 or 4, before the reality is it's so used up or messed up inside by its activity that the neutrophil will just die. All right. And then it becomes this dead cell that needs to then be cleaned up by the macrophages, right? The macrophages are not only patrolling for pathogens, the macrophages are like the custodians, right? They're cleaning up your tissue to keep it normal and healthy. And so now these macrophages are eating a ton of neutrophils. There's probably way more neutrophils arriving than they can consume. And so you end up with a lot of dead macrophages and neutrophils at that site. And that is what pus is. Right? Yummy, right? Nice discussion for after lunch. But that's what pus is. Pus is a ton of dead white cells as a result of this induced response. But hopefully, right, it worked. Hopefully, even though it might be kind of nasty and you have some pus, left over in the inflamed cut, right? That's all part of beating the infection, and then eventually your cut will heal. Right. Here's a question. How do neutrophils, or any cells for that matter, that are flowing around in your blood, like when they're released by the bone marrow, how do they actually know that they've arrived at the site of an infection? Think about it this way. I often try to picture, if you were a cell cruising around inside of your body, in your blood, what would you see, right? You just basically be in a tunnel, right? And the walls of a blood vessel, whether it's a large blood vessel or a small vessel, it's just endothelium. It's just the specialized epithelial cells that line the inside of your blood vessel. So you're rushing through this, and it's like a tunnel. And maybe you go from big tunnel to a smaller tunnel. Maybe there's a fork in the road, and you go smaller, smaller, smaller. But how do you actually know where you are, right? So this is actually a photo of, I think many of you know I grew up in Boston. And so the airport, Boston's Logan Airport, is actually built on kind of like this big landfill sandbar out in Boston Harbor. So it's not actually connected to the mainland, right? So in order to get to the airport, you have to drive through this tunnel or tunnel system. So they actually dug underneath the harbor, and you have this tunnel that you, on one hand, when you're approaching from the land side, you see the signs and you enter the tunnel, and then you go through the tunnel, and hopefully you know where you're going, because in the tunnel, there are different exit points. Right? And hopefully you have to know that you're actually under the airport and it's time to drive up and get out of the airport. Right? I think that's kind of amazing. As a kid, I loved going through these tunnels. Here in Hawaii, it's even more impressive because our largest tunnel here is probably like the H3 tunnel, right? And that actually pales in comparison to this. because it's one thing to go through a mountain, but this is actually going underneath the harbor, right? So there's seawater and fish and stuff above you. Anyhow, how do you know? Well, just like in this tunnel, this is the Sumner Tunnel that gets you to the airport, you have to keep an eye on signs, right? So in this case, you reach a fork in the tunnel, you either continue going straight and eventually you're going to emerge past the airport back where the tunnel leads back to the mainland, or you look for the sign that says Logan Airport, right? Exit 26. So now you know to steer your car over there. That's what happens in your body. In other words, the lining of your blood vessels at particular spots where there's an infection going on will display certain signs so that immune cells that are rushing by can see those signs and know to stop. So what are these signs? Obviously, the cells are not looking at something and reading something. These are particular proteins, right? Particular proteins that are the right shape so that proteins that are on the surface of the cell that's rushing by can actually join up. And that's how the cell knows it's arrived somewhere. Here are some examples of these different types of neutrophil endothelium adhesion molecules, right? We call these adhesion molecules because they stick together. So here on the right is a neutrophil, and on the surface of the neutrophil, there are these special proteins known as vascular addressins, or CD34. I like the name because vascular means it has to do with something that's going to happen in the blood, right? And addressin, yeah, right, it's an address. This helps the neutrophil know as it's going through your blood that it's arrived at a particular address. As part of this receptor or of this protein, you have these different sugar groups on it. These are what we call Sialo-Lewis X sugar groups. You don't really have to know that. That's kind of very O-chemy. But the point is, if you look, it's 3 sugar rings, and the point is the middle one is oriented kind of differently. This is a 5-ringed sugar compared to the 2 6 ones. The endothelial cells, the inner wall of the blood vessels, can express a particular protein known as a selectin. So that's what this pink thing is here. And that selectin has the right shape to be able to pick up on these special sugar groups. So as the neutrophil is rushing by in the blood, if the wall of the membrane or the wall of the blood vessel, suddenly you're entering a part of the tunnel that's coated with these selectins because the endothelial cells are expressing it, right? The neutrophil literally can start sticking, and that'll slow the neutrophil down. Okay? In addition, there are other interactions that help really stop the neutrophil. So that involves things like in this illustration here, you know, in your book, this is all the same illustration. I've changed it. I've cut it in half because the book did sort of a weird thing here. They actually switched the labels. On the bottom panel, the gray part is the neutrophil and the yellow part is the endothelial cell, right? That's kind of annoying, but whatever. So I cut it in half to make it easier for you to understand. The neutrophil also has these proteins known as integrins like LFA1, and those will bind to very strongly these proteins that are expressed on the surface of the endothelial cell known as ICAMs, or immunoglobulin-like molecules. And these interactions are very strong, and they really anchor the neutrophil onto the inside of the blood vessel wall as the first part of the neutrophil then beginning to undergo diapedesis and leave the blood and go into the tissue. So let's look at this here. This is a typical scenario where the white part of this diagram is the lumen of your blood vessel, right? So this is the inside of your blood vessel where all your blood and the cells and stuff are rushing by. And this is just one wall of the blood vessel, the basement membrane, and these purple cells are your endothelial cells, right? So, I mean, these would wrap all the way around. So at the top of the slide, you'd also have the other side, right? Because it's a cylinder that's cut in half, and we're just looking through the middle. Because there's an infection going on, these endothelial cells express things like selectins. And on the surface of the neutrophil, it has these addressin molecules. So as the neutrophil comes zipping by, it begins to stick to this part of the blood vessel wall. And the neutrophil actually begins to tumble. They've taken real video footage of neutrophils coming past a spot in the blood vessel where there are a lot of these selectin signs. And it really is just like a tumbleweed, like in the old Western movies, the tumbleweed blowing through the desert. The neutrophil tumbles, 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 tumbles. It's also kind of like, when I was a little kid, I had this, maybe some of you had this little rubber octopus thing that was like really sticky. It was kind of fun. And you'd throw it against the wall, right? And it would just kind of like stick and roll slowly down the wall, right? So that's what these look like. And eventually, the neutrophil comes to a stop, right? And so these selectins then are only expressed on the surfaces of these endothelial cells on the inside of the blood vessel in response to there being an infection nearby. So what is triggering these endothelial cells to even advertise these selectins, right? These cells are responding to something that's coming from the site of the infection. What is it? Oh, not surprisingly, it's those cytokine messages that the macrophages have been sending out, right? So here are some examples of different cytokine messages that are sent out by macrophages. We will study these over the course of the semester. I'm not going to go through all of them today, but very briefly, there are certain ones that stimulate inflammation. So cytokines that stimulate inflammation are known as interleukins. Okay. Again, you can take the name apart because early researchers saw that these particular types of cytokines were meant to go between white blood cells or leukocytes. So they're interleukocyte communication. So they called them interleukins, but then they realized they cause inflammation. Right? Today, let's just, for the sake of time, just talk about this one here, CXCL8. Right? CXCL8, the blue triangle here, recruits neutrophils from the blood and guides them into the infected tissue. So this is what CXCL8 looks like. Remember, they're just small proteins, right? And it's called interleukin 8. That's another name for it. The reason it's called CXC is because they typically have this type of amino acid structure or sequence where you have these two cysteines in the middle with something else in the middle, like a valine, right? And these cysteines can form disulfide bridges with other cysteines, and that's what gives this little protein its shape. So here's the ribbon diagram. Here's the three-dimensional space-filling model, small little protein, right? That's going to not only recruit neutrophils, but stimulate the neutrophils to move through the tissue towards the site of infection in a process that we call chemotaxis, right? Chemically induced movement of these cells. Once the neutrophils come to rest, right, you can see here, this was a tumbling neutrophil sticking to the selectins. It gets really anchored in place now because the neutrophil also has that other type of integrin, LFA1, anchored in place by the ICAM that the endothelial cell is expressing. Then what happens is the neutrophil begins to smell the nice smell of the CXCL8, right? And when I say smell, I just mean it has the right type of receptor, a CXCL8 receptor, that then binds to it. And what it does is that stimulates the cytoskeleton of the neutrophil, especially in this particular region where the receptors are being stimulated, to actually change, and the cytoskeleton begins to move in that direction. That's what encourages the neutrophil to squeeze its way out between the endothelial cells. That's the process of diapedesis, and it continues to follow its nose, so to speak, towards where the higher concentration of this CXCL8 cytokine is coming from, thereby leading the macrophage through its cytoskeleton constantly changing to move in that direction to the site of infection. Okay, just a couple more slides really quick, because I know we're already out of time. So I said that neutrophils are very effective as phagocytic cells. The reason they're so effective is because they have all kinds of enzymes in their vesicles ready to digest things like bacteria. These enzymes, though, if they were always active, would just digest the neutrophil itself, right? So these enzymes are oftentimes made as what we call a proenzyme. In other words, an enzyme that has a particular shape where it's not active, right? When the neutrophil begins to eat bacteria, it changes its metabolism very rapidly in a process that we call a respiratory burst, right? It undergoes aerobic respiration really, really rapidly, and that leads to a dramatic change in the pH of the inside of the neutrophil. Okay, it becomes a lot more basic first, and then it shifts to becoming a lot more acidic. And that change in pH causes these proenzymes to change their shape, and then they become active and they destroy the bacteria. As a result, both the pathogen and the neutrophil, right, are compromised, because the neutrophil, in this act of increasing its respiratory burst, it's changed its pH, it's messed itself up, right? So it itself is going to die after eating a couple bacteria. But the neutrophil has one more trick up its sleeve, right? And this, I think, is really awesome. One neutrophil eats maybe 3 or 4 bacteria and then it dies, right? But in the process of dying, what happens is the neutrophil will burst. And when it bursts, it spews all of its genomic material out. Right? This genomic material is chromatin. It's covered in sticky proteins. Right? It's a very sticky net that gets spread. And the sticky net can then trap many more bacteria. And in the process of exploding, this net is also carrying with it those activated, highly proteolytic enzymes that the neutrophil was using to digest the bacteria. And so you end up not just killing the 3 or 4 bacteria that it ate, but you kill dozens of bacteria that are caught up in this neutrophil extracellular trap or net. Okay, so it's almost like a kamikaze pilot, right? I think about it that way. The neutrophil is going down, right? So it might as well do one more act to try to inflict as much damage to the surrounding bacteria as possible. And then this is just a slide that shows you a site of infection. And all of these little purple dots are neutrophils, right? Neutrophils that are flooding the infected tissue, attracted there because they flowed through the blood. So this funny squiggly thing is a cross-section of a blood vessel that's kind of cut at an angle. That's why instead of looking round, it's kind of oblong. But these neutrophils came from there, were attracted by cytokine messages, came into the tissue to help out. Okay, sorry for going over, but let's go ahead and stop there today.